First, while you guys are writing some things down that you may need help with, I wanted to tell you just about a couple of my core concepts of anatomy and physiology. If you've watched my other videos before, core concepts, I like to think of these as just big picture ideas. And a lot of the times when you are learning A&P, you feel like everything is just coming at you at once and you have no place to put all of this new information. And so what core concepts do, for example, I've got a core concept, pretty easy one called high to low. And just think of this as like a little box. And anytime you see something in A and P going high to low, you can kind of add it to this box. As an example, we know that we have our lungs, right? So here are our lungs, kind of look like this. And inside the lungs, we've got our alveoli, little air sacs inside of our lungs. If we zoom in on those, what they would kind of look like is the following. We would have very thin, flat cells. If you know what those are called, drop it in the comments. And then we've also got the capillaries. So these are pulmonary capillaries, just came from the right side of the heart. They're carrying some blood, and obviously same cell type. Now we know that in our air that we breathe, about 21% is oxygen, and very little, about 0.001%, is actually CO2. Now, what's fascinating is in your body, you are constantly producing CO2. So CO2 will be relatively abundant in the bloodstream when it gets to this point. And oxygen, the gas, will actually be relatively low at this point because your cells have used up all the oxygen. They've been burning it up, making some ATP energy, right? Now, at this point, we need to ask the question, well, what's the concentration of CO2 and oxygen compared to this other location? That's another core concept, actually, is location matters. Location matters a lot. Okay, so as this example goes, we know that in the lungs, we've got a lot of oxygen coming in from the air. So since oxygen is a very high abundance inside of the alveoli, it's going to flow what? High to low concentration in the blood. That's how we saturate our blood with oxygen, gets picked up by our red blood cells. And at the same time, we've got the CO2 going high to low out into the air and then pushed out of our lungs and out of our body. So that's one example of how two core concepts, high to low and location matters, right? Two different locations, blood and alveoli, can come into play and really help you understand difficult concepts in a very simple way, which is what I'm all about. Okay, looking on the comments, I'm seeing hello from NH. I'm assuming that's New Hampshire. Otherwise, I have no idea what NH stands for. Nice. Thanks for coming back on. Sound check sounds solid. Thank you very much. I work on my voice beforehand. I needed to do some warm-ups. Learning about the tissues in skin. And then you said exam on chapters 6 through 9. What's the best study method to tackle this? Well, it depends on what, can you, I see a Sharika Knight. Can you tell me what chapters 6 through 9 are over? Because depending on the textbook that you're using, it might be different topics. Then I've got last semester, well done. On my final A&P course, anything to do with digestion would be appreciated. Also preparing for the T's. Miami is the best, good for you. Okay, New Hampshire, perfect. Good to know, Maria. Following from Zambia, awesome. Okay, what's the best way to study? Because darn, I'm censoring you. Can't watch a video without falling asleep. Well, if you're in a dark room, that'll probably make you want to sleep. So put the lights on, that'll be my one tip. Okay, so we got muscles, joints, skin. We can totally do that, y'all. Let's start with that. And then obviously I hear digestive system too. So we'll see if we can get some of that in as well. So the hard thing about this that I'm going to try to simplify is that depending on your teacher, they may emphasize different things more than others. So I'm going to emphasize the things I know is important in my classes, and then maybe it'll translate to what your teachers are giving you. So let's first start with that, I think you said joints, skin. So let's go into some skin first real quick. And this is relevant for everybody because the skin obviously covers your entire body. It's your biggest organ, so it must be important. And if we were to cut the skin and look inside of it, kind of like a layered cake, we would see three main structures. We'd see the epidermis, we'd see the dermis, and then if your teacher determines this to be a part of the skin, which some don't, we have the hypodermis. So let me write all those down. Hypodermis, okay? And then we've got the dermis in the middle. You guys should know this at this point if you've studied this. And then on the top part, you've got the epidermis. Now, this is a great example of how Obviously, you've got the same suffix, derm, like dermatology, always refers to skin, hypo meaning below, and then epi meaning upon or on top of. So think about it. If you were to guess, which of these layers are you actually looking at when you're looking at your own skin? Like what can you see? Well, obviously, we know that this is superficial, right? So there's another term, superficial, close to the surface. And then down here would actually be deep. So we're cutting basically into your skin. So if we're looking at your skin, we're looking at the epidermis. Now, if you think about it, this is the layer that comes into contact with your outside environment. So if you were to guess, I know some of you said I'm studying tissues, what tissue type do you think would be most beneficial to put in the epidermis? Would it be like muscular tissue? Like, do you need to move your skin like some weird skin monster? I wouldn't recommend that. But these epidermis, this is made of, we know, stratified squamous epithelial tissue. So we've got very thin, flat cells. And then at the bottom, we've got basically cuboidal cells. That's kind of interesting. So let me talk about the epidermis a little bit because I know it has a lot of other connections to other topics. So we know on the very bottom layer of the epidermis, this is actually called the basal layer, and these are considered stem cells. Now, why am I talking about this? This is going to relate to you, person who's talking about digestive system, because stem cells are always, always, always located in epithelial tissue. This is incredibly common and very important for all of you guys to recognize because think about it, epithelia. Think of that word, epithelia. It means, again, epi, upon, okay, upon something, and then thelia means sheet-like. Okay, so think of, you are literally taking a sheet of cells, in this case, right? That's what tissues are, multiple cells coming together for a common purpose. You got this sheet-like group of cells, putting it on top of a structure of your body. So this is how I always remember it in my NP classes. Everybody take your finger. I want you to touch a part of your body. Obviously, you can touch the skin. Obviously, you could open your mouth, touch inside here. You could touch your eye. You could stick your hand through your intestinal tract. And guess what? You're still touching epithelial tissue because it's literally the linings of your body. And what do the linings, right? What do the linings of your body come into contact with? What does the lining of your skin come into contact with? The outside world, right? What's really trippy, y'all, I want you to think about this, especially for digestive system. Think of your big body as just a 30-foot long tube of the digestive tract, right? So let's say this is like your mouth right here, and this is your anus or your anal opening, right? Okay, anus, if I can spell words while I speak. You've noticed that in my other videos. I can't write and talk at the same time. Now, one core concept I really want you to notice is location here. Think of this. This is your mouth, right? And then throughout the rest of your body, you've got like your skin, okay? And so think about, this is all epithelial tissue. This is a terrible drawing. But if this is like your skin, this is outside world, right? If I put food inside of my body, like here, is this really inside of my body? Think about it. So this is food going into your mouth, going down all the intestines, so the stomach, the small intestine, the large intestine, out through the rectum and the anus. Did it ever go, quote unquote, into my body? Well, you know that some of the food would be absorbed. Now, if we absorb food along that digestive tract, it basically means we're taking the nutrients and throwing them into the bloodstream. So we're chopping these up and we're throwing them into the bloodstream. I want all of you, this is so important for A&P 1 and 2. Remember that anytime something is in your bloodstream, it is considered inside your body. Okay, it's inside your body. Why is that? Well, another core concept I know is the blood is a river of life and it travels to virtually every cell of your body. So if you get something into your blood, it could virtually go everywhere and affect any cell of your body. Okay, vitally important. Now, back to my original point. Is this food that you tried to digest, if it went out of your body, did it ever get into your body in the first place? No, actually, this is technically outside of your body. The inner lining of your digestive tract is outside of your body. Only if you put that into the bloodstream will it be considered inside because you can transport it now to all the cells of your body. Now, back to original point. We know that the epithelial tissue is lining all of these structures. Think about it, y'all. Your skin gets beat up on a day-to-day basis. I mean, if you're wearing this awesome organized biology shirt that you can buy in the shop below, it's rubbing up against my skin constantly, constantly, constantly. So my skin cells are actually getting whacked and damaged and killed potentially. So some of these skin cells will actually slough off as they get whacked. Furthermore, you're putting rough food into your digestive tract. There's a hydrochloric acid inside of your stomach that could damage some epithelial tissue. So all of these epithelial tissues need to be able to what? Regenerate. So know that the epithelial tissue most of the time will be very regenerative. So therefore, what cell type will it have? Some form of stem cells. By definition, stem cells are cells that can basically divide and differentiate into some other cell type. As this example goes, you get the basal cells, they will divide, okay, upwards, and eventually they will form keratinocytes. Now for the integumentary system people, these keratinocytes, look at that word, keratinocytes. Keratin is a strong, tough abrasive protein, so it protects you against damage. And then cyte, that means cell, always. So anytime you're thinking cell, it's just one of the functional units of life of your body. Okay, you got 30 trillion of them. They're very, very, very needed. So we got basal cell, stem cell, differentiating, dividing into keratinocyte, a different type of cell that will eventually get pushed up, and then sloughed off. Now, another key point that I was talking about, remember blood is inside of your body. Well, think about it. The blood is only in the dermal layer as well as the hypodermis. So blood is going virtually all over the place here. Okay, blood is a river of life, goes everywhere. Why do these cells die then? Why do these keratinocytes end up dying? Why do they slough off? Well, it's because blood is a river of life. And if blood doesn't touch a cell, right, if we don't have a connection to it, those cells will die. Always remember that. That is true too of if you're taking A&P2. I saw one person's taking like a second semester class. The heart, right? Heart attacks are essentially when the coronary arteries, the arteries that feed the heart tissue, get blocked. In the blood, therefore, gets blocked. And so there's not blood getting to the heart tissue. If there's not blood getting to the heart tissue dies. Okay? So a couple other things in the dermis. The dermis is mainly made of dense, irregular connective tissue. It's strong, kind of stretchy. We've got some collagen fibers. We got some fibroblasts. Fibroblasts meaning fiber builder cell, fiber building cell. And they will basically regenerate the dermis, especially if it's damaged. We've got some erector pili muscles that can hold and pull that hair follicle that will be like stretching outside of this part. There'll be sensory neurons so we can detect things in your skin and feel things. And then obviously we get the hypodermis. This is made of primarily adipose tissue. So adipose tissue, that's just fat tissue. And adipose tissue is funny because it's literally these fat cells. Like the cells look really fat. The nucleus is pushed over to the side. And this is mainly for energy storage, insulation of heat, as well as water. We don't want water getting out of our body, evaporating and drying us out. So one connection here clinically, if a person's actually starving, like they don't get enough nutrients, this adipose tissue will break down as energy, and then the dermis itself will stick to underlying structures like bones and muscles, which is why if you've ever seen like really like war movies, like I'm thinking Hacksaw Ridge, like those are sketchy movies. Like if that guys go into those internment camps and they starve or nearly starve to death, their skin just sucks into their ribs and their muscles. And that's why you can see it, because the adipose tissue is being broken down for energy. So I'm going to pause there. Let's look at some of your other comments. Let's see what you got. Okay, so we've got anything to do with muscles and joints. Okay, McGraw-Hill's the book. This topic is actually part of chapter 6. Hey, perfect. Hopefully this has been helpful. Drop some other things. I'm going to go into, what is the difference between cilia and microvilli? Good question. So cilia, let's go into that real quick, and then we'll get into maybe some muscles and joints perhaps. So microvilli, let's actually keep this model up real quick. And on the intestinal cells, specifically, these are pseudostratified columnar epithelial cells. Big, long word that just means they look like they have multiple layers of columns, but it's really only one. Now, lining them, let's say this is in the small intestine. Okay, so here is the small intestine. We're going to zoom into it. So here's the cells of the small intestine. We will see, okay, in the cells themselves, there will be these little projections, okay? And they'll all be kind of lined up next to each other like this. Okay, it kind of looks like if anybody's a SpongeBob fan, the glove world, when they had like those gloves out. So here are the cells. The cells have projections on them. These are literally translated to microvilli. I think that means tiny finger, if I'm not incorrect. So microvilli is essentially the same thing structurally as cilia, but cilia is found in a different place and has a different function. So let me give you an example. Microvilli, what this does is increases surface area for absorption. Because think about it. At this point, these are intestinal cells wanting to absorb food into the bloodstream from the lumen or the inside lining of the intestinal tube. So think about it. If we've got all these nutrients flowing through the tube of the intestines, well, these guys can enter here, they can enter here, they can enter in here. They've got all this surface area, right, to absorb this food into the cells and then pass into the bloodstream. So those are the roles of microvilli. They're basically, I would almost consider them similar to cilia, but they're not going to like move, okay? So let me give you an example for cilia. We talked about the lungs earlier. So we've got the tracheal tube. Oh, that was a terrible drawing. Here's the trachea, and that's going to branch off into bronchi and bronchioles, etc. So this is in the lungs. Lining this tract, okay, lining this tract will have cells, okay, on both sides that again will have these little branches extending off the cells, okay? Why is that? Well, in this case, we're making first off some mucus. So some of these goblet cells, they'll kind of be shaped like this. They'll be spewing some mucus into the area, lining this tract. And what happens is essentially pathogens will get in through the right tracheal tube. So here's some pathogens out here, so like viruses, whatever, and they get in here and they want to infect your lungs down here. But instead, they get trapped in the mucus. Well, if they get trapped in the thick mucus, we need to not let them go down into the lungs. So these cilia will actually push their projections that way, right? And you've probably had this happen before where you have like something in your throat and you're like, right? That is because these cilia on these respiratory cells are, they're technically, I believe these are just columnar epithelial cells that have these little branches. Or no, these are pseudostratified. They're pseudostratified because they've got the projections. And they'll start pushing it upwards, right? So it's basically the same structure, but two different functions. One's for increasing surface area, one's for increasing movement of the mucus out of the respiratory tract so that you don't get infected. So I'm thinking we're going to go into let's see here. We're going to go into muscles, and then I'll maybe give you some helpful study tips for it too, for you who is wondering about how to study properly. So I also saw that question on, can you explain what extracellular matrix is? That's basically the space outside of a cell. So extracellular means outside the cell. Matrix refers to the fluids and fibers that are comprising the space outside of the cell. So just as a really quick example of dense irregular connective tissue, we talked about the fibroblasts right here. The fibroblast stands for fiber building cells. Fibroblast, fibroblast. They will lay down the proteins, collagen, in the space here, right? But then there will also be this space filled with fluid actually from the blood plasma. So there's going to be some fluid in here too. So we see how we have cells, gels, fluids, and fibers. The gels and the fibers comprise the extracellular matrix, the space outside of the cells, whereas the intracellular part of the cell is inside of the cell itself. So hopefully that's helpful. Now let's talk a little bit about maybe some joints and go into that. If you guys have any other topics, again, drop it down there. And if you want to like and subscribe to this channel, maybe donate on that little comment thread, feel free, but I by no means expect it. In the future, however, in the description below, I also have a Calendly invite. If you want to do personalized one-on-one study sessions with me, you can schedule it there. I've only got a little bit of availability, so make sure you sign up for it. I'm going to try to extend that time later on. So let's get into muscles, one of my favorites. So first off, let's think about the muscular system, okay? We've got several different muscle types, and we're primarily going to talk about the one that I know you guys are talking about. We've got our skeletal muscle, which is the main one we're talking about. We've got cardiac, and then we got the smooth muscle. Now, I always remember to make it simple, remember muscular, M. Anything that moves in your body is going to be moved by muscular tissue. So think of the intestines, they're contracting, peristalsis. That's done by smooth muscle. Your heart is constantly contracting, pushing blood. That's cardiac muscle. Skeletal muscle is basically moving your skeleton with the muscles attached to it. But to make it even crazier, if you look at my pupils, if I were to like close my eyes and then open them, my pupils kind of constrict pretty quickly. That's also done by muscular tissue, smooth muscle particularly. Okay, so what makes skeletal and cardiac similar is that they are both striated. So in other words, if you look at the muscles themselves, you will see basically a bunch of stripes, striations. That's why they're called striated. Now, I make this simple. I know it's not perfectly anatomically correct, but I think it is. I think in order to contract this muscle, think of this muscle, the striated muscle, as an accordion. Have you seen an accordion before where they have all those stripes? How do you contract it? You have to bring the stripes together and then extend them away. So that's essentially what's happening here. In order to contract this muscle, just bring those lines closer together. And then what do we have? Well, we have those three lines, but now they're closer together. We have a contracted muscle. So that's the whole goal of striated muscle, bring those lines together. We'll talk about those in a second. Now, the main difference you guys could probably guess is that skeletal is voluntary. We have regions of our brain that can actually innervate by multiple neuron system this voluntarily. So it's a conscious thought. I want to contract my bicep. I can think about that and do it. Whereas cardiac and smooth, we're talking about the intestines and the heart. These are involuntary. They're controlled by other regulatory processes that we don't have direct conscious control over. Make sense? Okay, and yeah, I think that's what I wanted to talk about there. Now let's talk primarily about skeletal muscle. You got I believe the number is 550 skeletal muscles in your body. The main goal is for movement, but they're also producing heat because we're going to be breaking up some ATP, releasing heat as energy. And also, yeah, just movement of blood, actually. We've talked about in the cardiovascular system, you can increase venous return by contracting your skeletal muscles, whole lot of functions with muscle. So let's first start, think about a whole muscle. So I'm going to see this big old muscle here. Okay. And in reality, we would attach that muscle to a bone. Okay. So I'll just draw a bone here and then a bone here. Obviously that does not, it does not look exactly right. But you'll see at the very tips of the muscle, we will always have tendons. Okay. These tendons are going to connect muscle to bone. In reality, anytime we're talking about a joint that we're going to be moving, so two spaces between bones, the bones will also have ligaments connecting bone to bone. So these will be the ligaments. Now, what's cool about these two structures is that they are both the same tissue type. They are called dense, regular, connective tissue. I like to think of regular as a rope. It's a very strong structure in one direction, and it does not stretch. So think about it as like a rope. It's got like all these intertwining collagen fibers, super strong in one direction. You may have some fibroblasts around here building that matrix. But these tendons are so strong, like the Achilles tendon can take up to about 2,000 pounds of force per square inch. Really strong rope-like structure. But again, they can't stretch. Muscles are able to extend and contract, but the tendons and the ligaments themselves don't stretch. So if they get stretched, they become taut and they try to hold the bones together. But if you put enough force on them, as we've seen with many football players, soccer players, put enough force on them, they just rupture, and that actually requires surgery. They don't heal very well. Now let's talk about the muscles themselves. If I were to cut that muscle open, okay, I would see a bunch of really subdividing structures. So we're going to look inside of this and imagine that we are looking inside of the muscle. We would see several different fascicles, okay? So these are fascicles running through the main part of the muscle tissue. I'm trying to imagine how I usually draw it. So just give me a second. So these are muscle fascicles running through the entire length of the muscle, okay? So let's see if I can kind of draw that like behind it, and behind those guys, and then behind that guy. Okay, and they're running through the whole muscle. And again, let me write the word fascicle, muscle fascicle. Now you'll notice in between, there's some space here, right? There's some space kind of connecting the two, okay? And I would actually refer to this as the fascia, right? It's surrounding, okay, the muscle fascicle. I believe it's called the endofascicular layer is what I call it typically. But anyway, they're just holding these fascicles together. Or shoot, I haven't looked at this chapter for a while. I think it's like either peri or endo surrounding it. So I apologize for that. But anyway, inside the muscle fascicles, more importantly, we will have several different other subdivisions. Okay. These subdivisions that will come out, I'm going to zoom in on as well. These are going to be the actual muscle fibers, okay? So here we have a muscle fiber. Now I want you to remember that fiber is a cell. It is a muscle cell, but it's called a muscle fiber. Kind of weird, right? Now in the muscle fiber, since it's a cell, we'll see a variety of different organelles. So we will see a variety of different nuclei. It's a multinucleate cell, skeletal muscle is. And inside of that, we'll have several other very important structures. One will be, I'm going to draw this kind of labyrinth of a variety of different structures here. Okay, and this is actually going to be called the sarcoplasmic reticulum. Sarcoplasmic reticulum. Okay, let me point to that. Sarcoplasmic reticulum. Okay, something here. Anytime you see sarco, that refers to it. Actually, I think the Greek is flesh-like. But I always just refer to it as muscle. Anytime you hear sarcomere, sarcoplasm, all these things, sarcolemmic hypertrophy, it's all referring to muscles, okay? Now, reticulum is kind of an intertwined structure within the muscle fiber itself, but I want you to mainly know that it stores calcium ions. If you remember nothing else from this, calcium is the key to contraction. Calcium is the key to contraction. So if calcium is released from the sarcoplasmic reticulum, I'm going to pretend it's inside of here. If it's released into the space, into the cytoplasm of the muscle fiber, I believe it's called the sarcoplasm, not to be confused with the sarcolemma, which is the plasma membrane. If the calcium gets out, we can contract the muscle. Why? We have, among other things, I'm not going to talk about them too much, we will have overlapping thick myosin as well as very thin actin filaments running alongside each other. Okay, so I'm going to draw kind of this like that. Okay, and connecting to the actin will be something called a Z line. Okay, on both sides, Z line to Z line. You'll notice that these Z lines kind of look like the striations, and in fact, you can actually see a very thin Z line there. And again, these are going to be actin, A-C-T-I-N, and then we've got myosin. Again, the thick filament. So thin filament, thick filament. Thin filament, thick filament. One fun thing here is that myosin literally translates to the muscle protein. It's going to be the one that quite literally looks like a muscle and is moving stuff. And then actin, fun fact, is again a protein. It ends in IN, but it is the acted upon protein. So myosin is going to act on the actin, right? Makes sense. So let's zoom in on that and see what's happening. First, before I do that, I want to point out that this is a Z line over to the Z line. So this is Z line here, or otherwise known as Z disc, Z line. And what's going to happen is we're going to pull these Z lines together to make the contraction. And that one unit of Z line to Z line is called a sarcomere. Again, translating to muscle unit of contraction. So that is, in a way, our accordion lines that we're going to bring closer together. So let's come over here and talk a little bit about muscle contraction. Okay, I see no new. Oh, thank you, Mel. You're so kind. Oh my goodness. Mel gave me $4.99. You're the best. I wish I could like star you or something. I'm not sure what to do, but I'm going to shout you out and you're going to live forever in this video. Thank you so much, Mel. You're so kind. Man, really didn't have to do that, but I appreciate it. Okay, now let's talk a little bit about what I like to call the sliding filament theory. Sliding filament theory. Now, one thing with this, well, two things actually. Sliding filament theory. Sliding filaments. We're referring to these two proteins, actin and myosin, that are going to be sliding across each other. Okay? Theory in anatomy and science always refers to we're answering the question why or how. So in this example, how do the muscles, skeletal muscles specifically, contract? How do the skeletal muscles contract? It doesn't mean that we are wishful thinking about this. No, we know that this is 99.999% sure that this is how it works. So let's learn about it. So I'm going to draw the Z line actually in black just to kind of differentiate it. Okay, so we've got Z line here, we got Z line here, and what we will have in the sarcomere, right, the space between those two, that unit, we will see a very thick myosin protein, okay, with little heads on it. Okay, so I'm going to draw a couple little heads. So these are still a part of the same protein, myosin, the thick filament, but they're called the myosin heads right there. And then we will see very, very thin actin filaments lined up like here and lined up like here. And these are in repeating units. So if we were to go down a little bit, we'd see the same thing. Sarcomere, we got myosin, actin, actin. And all of those together, if we pull it together, we can contract the whole muscle. Now, in order to understand how this happens, we need to understand what's going on in this particular space. Remember, myosin is going to kind of grab the actin, pull it towards the center, the M line. That's technically the M line if you need that. It's an imaginary line, but we've got M line here. And we'll also have some other proteins that I don't really point out too much. We've got titin. It's kind of like this big long spring that goes all the way from Z line to Z line. That's your titin. And then we're also going to get into a couple other proteins that we don't see necessarily on this level. Now, there's actually a theory that titin plays a bigger role in muscle contraction than we think, but I'm not for sure about that one, so I'm not going to go into the details of it. Okay, so we're zooming in on that little corner section. So we've got myosin heads, okay? Attach that thick myosin filament. We've got the actin. In reality, the actin would basically be these small circles. So I'm going to try to draw them as best I can. It'd be these small overlapping circles. So you kind of have one on the bottom, one on the top. Kind of looks like a nerd's rope, in my opinion. Okay. And that will be attached again to that Z line. That will be here. And what we need to have happen is the myosin needs to grab the actin and pull it inward. So we say that on the actin, there are these little sites. So I'm going to draw some reds down here. And these are going to be called myosin binding sites. Very easy remembrance, right? Sometimes we're nice with our protein names, right? So this is where myosin is going to grab actin, but there's a problem. There is actually a protein that is covering up those myosin binding sites on the actin. So I'm going to draw this protein here, okay? And this is called tropomyosin. Tropomyosin. Now, the name tropo usually refers to it's acting upon something. It's not really acting upon myosin. It's actually kind of acting more on actin, but it's covering the myosin binding site. So that's what we're going to write. Covering myosin binding sites. So right now, myosin cannot grab the actin. Myosin cannot grab the actin, okay? But we know that tropomyosin, big long word, big long chain, is connected to some different proteins, okay? So I'm going to draw this protein connected to tropomyosin, and this is called troponin. I want you to remember troponin is kind of like the lock, okay? Troponin's kind of like the lock. Why? It's bound to tropomyosin, which I want you to remember as the chain, okay? Big long chain, big long word, lock. Troponin is a very short word. You can kind of remember short word lock, short word troponin, long word tropomyosin, long chain. Now, what did I mention earlier is the key to contraction? Well, calcium right there, right? So calcium, when we want to contract the muscle, this is what is fascinating. At this point, we are not contracting the muscle because myosin can't bind to actin, yeah? But as soon as we send a nerve impulse, a voluntary nerve impulse down into this neuromuscular junction, we don't have to talk about it, neuron talking to muscle, that muscle cell will tell essentially its sarcoplasmic reticulum to release calcium ions into, again, all this fluid so that it can interact with these proteins. So I'm going to draw calcium like this. And what did I say calcium is the what? Calcium is the key to contraction. So as soon as we get calcium, calcium will bind to the troponin. And what will happen is the tropomyosin will get pulled off of that location matters. And it will actually just kind of pull back over the top of the actin. When this occurs, you can predict that the myosin's like, hey, my friend is here, right? I can grab my friend. And that's exactly what will happen. Myosin, I'm going to draw it kind of coming up. Myosin's going to bind to actin, okay? Now, it won't extend like that. In reality, it's closer, but I drew it kind of far away. Myosin then binds to actin. And what will happen is that myosin will do a power stroke where it will actually drop, and I'll teach you about this in a second. It'll drop off the ADP and a phosphate that's already been cleaved off. Those will pop off, they will leave, and they will complete a power stroke where the myosin pulls on the actin that way. And when we do that, once again, we pull on the actin, we pull on the Z line. We pull on the Z line in both ways. We shorten Z line to Z line, we shorten the sarcomere, we contract the whole muscle if this occurs, right? And that's why it's called the sliding filament theory. The actin is literally sliding across the top of myosin, the thick filament, okay? Now, what's up with this ATP thing? Let me briefly talk about this. This is probably the most misunderstood part of muscle contractions. Let me erase this a bit. What happens constantly, like right now, you're either sitting down, standing up, laying down, your muscles are relaxed, but they're still generating a lot of our favorite energy currency molecule, ATP. Now, if you're going to do any sort of work, any sort of movement, we need to use ATP. Now, the way we use ATP, this is going to get back to your chapter 2, chapter 3 in chemistry. We've got ATP, stands for adenosine, and then we've got a triphosphate, okay? 3 phosphate groups, that's why it's called triphosphate. Now, what's really interesting is these are covalent bonds between the phosphates. Just basically means there's electrons being shared between them. There's energy stored there, but primarily in this last phosphate. I want you to imagine that there's just crap ton of energy stored in this last bond between the 2 phosphates. So what will happen is the myosin, interestingly, has an enzyme called a myosin ATPase enzyme. Well, that stands for an enzyme that's going to break apart what? ATP. So when myosin sees ATP, as you're just chilling, your mitochondria are making a bunch of ATP because you've eaten glucose and oxygen, all that stuff, you've got plenty of this. We are going to clip that phosphate off. The phosphate will go flying. What's really interesting is 2 things occur here. Anytime the phosphate goes flying, we know it's going to do some sort of useful work for the cell. Okay? What's that work going to be? It's actually going to recock the myosin head. I'll talk about that in a second. So it's going to be doing some useful work. But at the same time, we will also produce a byproduct of heat. So that is one reason why when you contract your muscles, you're using a lot of ATP. As you're using it, cleaving that phosphate, you're producing a bunch of heat. That's the biochemistry behind heat production as you're contracting your muscles. Kind of cool. Now, what's the useful work? I told you that that is to recock the myosin head. So say we just contracted the muscle, okay? And the myosin is now droopy and sad. Okay, droopy and sad. It is low energy, right? It just did the work. It's like a catapult. You let go of the catapult, now the catapult's just chilling like that, right? Has no energy. Well, when that myosin's sitting there, if ATP is present, right? If ATP is present, that ATPase enzyme will cleave that phosphate, turn it into ADP and a phosphate. And what will that do? We're doing some sort of useful work, right? What is that useful work? We throw that myosin head back so it is energized now. So right now we've got ADP and a phosphate bound to the myosin head, and it is energized. So I want you to think of this as an energized myosin head. So if that myosin head is energized, well, guess what? If myosin binding sites are present, we can do the same cross bridge and power stroke, right? And as long as ATP continues to remain present, that myosin can cycle. This is called cross-bridge cycling. We're energizing myosin with ATP. We grab it, we pull the actin, we energize myosin with ATP, we do it again. That's cross-bridge cycling, okay? So that is all about ATP being used in the muscle. And that's actually why if you exercise for long enough, right, you're depleting your ATP storages, yeah, because like you're using up all this oxygen, you're using up all this glucose, making ATP, but then you're depleting, your muscles start to get tired, right? That's where fatigue begins to set in. So hopefully that was helpful. All right, gonna read a few other comments. Welcome if you're just joining in. We've gone through, muscles, we've gone through skin, we've gone through digestive system. It's been so fun. Just getting into glands, if you have time to cover anything helpful about that, I can tell you a little bit about those. Anything to do with hormone oxytocin and how it promotes uterine contractions, what muscles are involved? Good question. Brand new A&P student, I've been really enjoying your videos. Very helpful. Thanks for doing this. You're the best, the land of many projects. And Sunday Muchika, thank you for being here. And then Uwera Susan, good to have you. Hallelujah. Praise God. He's good every day. So happy Sabbath. Okay, so let's go into kind of glands, hormones, oxytocin. Definitely get into that for you guys. We're just doing like a random, what do they call this? A fruit basket up set of topics. Pretty fun. So I'm grateful that you guys are here. Again, if you're just joining, this is a live stream. We're just doing some study topics for A&P. Ask me anything. If you want to do a one-on-one tutoring session with me, you can use my Calendly invite. It's in the description of this video. I have very limited availability, so definitely get signed up. It'll be just one-on-one Google Meet with me, so I can help you out with anything you need. And I also have some resources for like pre-nursing, nursing, all that stuff you can look at. And then if you want to buy a shirt, you want to support the channel, you can do super chat, whatever you'd like. But again, no pressure on any of that. I just love what I do, so I'm grateful that you're here. Okay, let's talk about glands. So first off, we got the cell type, right? Glands. The cell type will always be epithelial cells. Okay, why is that? These are glands that are lining your body, right? They're lining your sweat glands, right? They're lining the skin so that if they secrete something, it secretes out of the body onto your skin. You also have, we'll talk about the posterior pituitary gland is lining your bloodstream, so we can secrete things into your bloodstream. You might hear a similarity there. So glands are always epithelial tissue, but they're always going to be either cuboidal or columnar or pseudostratified columnar. So they're always going to be these sizes. They are never, okay, 99% of the time, they are never going to be squamous. Why? Imagine the space this cell has to make a product. Glands are basically secretory cells, right? Secretory cells, if I can spell cell right. A secretory to cells just means we're secreting some sort of product. We're producing a product inside the cell and we're going to spew it out. So as an example, let's say we've got this cell here and it's going to spew this product out of itself through exocytosis. Well, if you think about it, that cell has plenty of space to produce that product, package it into something called a vesicle, right? And then spew it out of the cell. Yeah? Look at this guy. This guy ain't got no space. It's like cooking in a galley kitchen in New York City studio apartment. There's no space. Your butt is in the stove as your feet are in the dishwasher. You have no space. So really, these cells will never be glandular. They'll never be producing a product like these guys. These guys got space for it, right? The cuboidal and the columnar cells. Now, that being said, let's say this guy secretes his product through, again, exocytosis, where basically the cell is producing something, packaging it in this little capsule, this vesicle, a membrane around it with a product inside, pushing it out of the cell, spewing it into this space. If this space is technically outside of our body, we call this guy an exocrine gland. Exocrine, meaning outside secretion gland, okay? Now that being said, I wanna clarify something that students get confused on a lot. You also have the stomach. So here's the stomach, okay? I mentioned at the very beginning of this video that the inner lining of the stomach where your food goes is technically outside of your body. So you will have certain cells, I believe it's the parietal cells, lining the stomach, and they'll be producing hydrochloric acid. They're a secretory cell producing hydrochloric acid. These are technically exocrine glands because they're secreting it outside of the body, because if you continue on, we are technically outside of the body, right? Now, exocrine means outside secretion. This guy we're going to say is going to be called an endocrine gland. Endocrine means within secretion. Now, where is inside of your body? If you've stuck around this whole time, we know that inside of your body is the bloodstream. And if it is an endocrine gland, we will again have vesicles. We will have a product, and that product will be spewed out into the where? The bloodstream. I'm going to say this again. 99% of the time, unless you're like the liver, these are going to be hormones, okay? So these are hormones. We're going to get an oxytocin here. These are chemical messengers that travel in the bloodstream and change cell functions change cells' functions to maintain homeostasis. Great. Okay, so the chemical messengers in that are going to go in the blood. We know that the blood goes everywhere, and they're going to change cells' functions to maintain homeostasis only if that cell has a receptor for that hormone. Okay, that's a key core concept. No receptor, no change. So, in order to respond to any messenger, any hormone, any neurotransmitter, we have to have a receptor for it. So, as your example, I want to make sure that I cover it. Oxytocin, how it promotes uterine contractions. Yes, totally. Okay, so what we have with childbirth in this case, and I'm going to get to, you said, can you go over nervous tissue? I'll do my best. Yes, it's pretty intense. I'm not sure how deep you want. So if you're asking about the nervous tissue, let me know, like, do you want me to go over neurons and action potentials or more like the glia and that type of thing? Okay, so back to oxytocin. So we know that we've got an endocrine gland called the pituitary gland. We got an anterior and a posterior part. So I'm going to say this is the anterior front-facing part, posterior backside part, and this is going to be the one that we focus on, the posterior. We've actually got these neurons down here that will release the hormones, interestingly enough, into the bloodstream. That hormone we're going to draw as a triangle is going to be oxytocin. Okay, now oxytocin, when is this released? Well, we know it's released, please forgive my drawing, during childbirth. So we've got, I'm going to have a mother here. Okay, this is how I always draw it in class. Here's the mother, very pregnant. You've got the baby pushing down, about to take a joyride downward. And he's pushing, he or she is pushing on a structure called the cervix. So the cervix is basically this closed, tough membrane that basically is closed like this. And as baby pushes down on it, it stays closed to keep it inside until it starts to stretch and start to allow baby out, right? So what happens is the cervix will begin to stretch, and you've got these stretch receptors. I'm going to kind of draw them in orange. Okay, little stretch receptors that will send a nervous signal back to the hypothalamus, which is actually up here. These are hypothalamic neurons. The hypothalamus is the main regulator of the H word, homeostasis. So these neurons start secreting oxytocin in response. Well, oxytocin travels in the bloodstream. Where does it go? Everywhere. And it's going to come and act on the structure that the baby's kind of sitting inside right now, which is the uterus. So here's the uterus. We've got the endometrial lining that the baby's been fed from with the placenta and all that. But then you've got the uterus with its muscular tissue. It's smooth muscle. Okay, so the smooth muscle of the uterus is wrapped around it. Just think about it as it's going to close, baby is going to get pushed out. So as it closes, baby pushes out. And it's actually more like it pulls up and out. So the uterus will actually pull up and out. So baby will literally just slide right out. It's kind of interesting. If only it were that easy, right? If it just slides right out. I know a lot of mothers, if you're watching this, you're like, it was not very easy. I get that. I'm not saying it was. So here's the uterus. What will happen? Well, oxytocin travels in bloodstream. Uterus has a lot of blood supply, but it will have these oxytocin receptors. When oxytocin comes in and binds to these receptors, it will actually increase, if you want to know the mechanism, it'll increase calcium ions inside the uterus. So therefore, the uterus will be stimulated to contract and begin, again, pushing and pulling upwards so that baby will get sucked out. Now, as baby pushes more and more on cervix, you can guess, more stretching, more oxytocin, more contraction, more stretching, more oxytocin, more contraction. That's called a positive feedback loop. We're amplifying that stimulus and we'll have the baby born. Now, that being said, you asked the question, what muscles are involved? Well, the uterine smooth muscles are the main ones that will push baby out. But you can't control those. These are involuntary contractions that are done by the hormone, right? And that's actually, if you've ever been induced, you get Pitocin, which is the synthetic form of oxytocin. And so as it acts on the uterine smooth muscles, pushes, you feel the contraction, but you can't control it. But the doctors and the nurses will tell you to push, right? Well, what are they telling you to push? Your skeletal muscles, the abdominal region surrounding the area, so that if you can narrow that by contracting your abs, like rectus abdominis, all the transverse, whatever, as you're contracting those, it'll decrease that space and help the uterus along. Now, the skeletal muscles themselves aren't the main reason baby gets out. The main reason is the uterus, but they do help a bit. Okay, hopefully that was helpful. If you need more clarification, please let me know. Will you post this video live on YouTube? Of course I will, Afia. Yes, just an overview is fine. This is going along with the other types of tissues I have to learn as well. Okay, that will be a good finish. Let's finish up with nervous tissue as an overview. And then, yeah, I think we'll sign off for the day. I'll post this so you can watch it on your own time. Thanks for being here again, guys. So, lastly, but not leastly, I always say that. It sounds stupid because it is nervous tissue. So let's just overview tissues real quick. You've got the epithelial lining the body. We talked a lot about that today. Then you get the muscular tissue. We talked about that. Then we have also the connective tissue that I don't want to go into because there's so many subsections and stuff. And it's a variety of different tissues like blood, bone, tendons, ligaments, all that stuff. So we're going to go instead into nervous tissue. Now, nervous tissue, the main part, okay, will be the neurons. So here will be a neuron. I'm going to draw it very poorly. And the neurons are essentially a signaling cell. So they are made to receive stimuli. So I'm going to say stimuli. The way they receive it is they have these little branches called dendrites. Okay. So previously, remember when I was talking about the stretch receptors in the cervix? Those were actually the dendrites of sensory neurons that detected that signal, that stretching. The same thing happens when you're like pulling your hair, touching your skin, slapping your face. Don't do that. But the dendrites of the sensory neurons are detecting that. Okay. Now, once they detect it, they will take that information and actually make the neuron negatively charged to positively charged very quickly. And that positive charge in the soma, the body of the neuron, will travel down the axon, long signaling branch, and it will send that positive charge down to the end of the axon. This is called an action potential. We've got a lot of videos on the nervous tissue and neurons that you can learn about that. When we send the action potential, it's going to communicate to some sort of target tissue, so like target cell. The cell will get basically a neurotransmitter. The neuron will release, cell receives the signal, does some sort of response. Okay, so that's typically how neurons work. Now, these neurons are found in a variety of different places. They can sense things, they can integrate things, and they can also coordinate motor output. So when we talked about moving the muscles, like right now, I'm telling a motor neuron to send signals to my muscles to tell them to contract. But we clarify that by saying, hey, some neurons are found in the central nervous system, which is going to be your brain and your spinal cord. And then there will also be neurons in your peripheral nervous system, which is going to be like your nerves, your sensory neurons, and your motor neurons coming in and out of the brain and the spinal cord. Now, in the central nervous system, there are a variety of different glial cells. Now, glia means glue. Glia or neuroglia are basically the glue cells. It looks like they're gluing the neurons together. In reality, not all of them do that, but that's why they called them glial cells. Now, in the central nervous system, we need to help out these neurons. So let's say this neuron is in the central nervous system. We'll have a variety of different glial cells that are going to help this guy function. One such one is something called an oligodendrocyte. So I'm going to draw this little wrapping around the neuron, okay? And we've got this glial cell called an oligodendrocyte, literally translating to many branched cell. So this oligodendrocyte creates a wrapping of its own membrane around the axon of the central nervous system neuron. This is called myelin, and it's like the insulation of a wire where it allows that signal to be sent very quickly, and it also protects the axon because these can be up to like 3 feet long in some cases, so that we really need that insulation to protect it, just like any cord that we have. So that's one. Another one that's really interesting are called these microglia. So microglia right here. Microglia, think of these as the immune cells of the central nervous system. So if you were to ever like get a concussion, as an example, you get whacked, your brain hits the inside of your bones, it doesn't feel very good. These microglia can migrate, so I always remember microglia migrate to damaged sections of your central nervous system and help basically coordinate the inflammatory response, the immune response to clean up the area. You'll also have other cells. I'm kind of losing some space here. Let me draw another one over here. So here's a neuron. Okay. You'll also have a blood supply coming to a lot of the neurons, but the problem with neurons is they're very, very needy and very, very sensitive. If they get some certain things from the blood, like metabolic wastes, from the blood into the neurons, you'll actually kill the neurons pretty quick. And you don't want that, obviously, because your neurons kind of keep you alive. So you will have these amazing, amazing cells called astrocytes. They literally translate to star cells, okay, because they kind of look star shaped. So I'll kind of draw that. Okay. And these astrocytes, there we go. These astrocytes form what's called the blood brain barrier. Blood brain barrier. What is that? Well, the astrocytes are kind of like the bodyguards of what is in the blood. So say there's some sort of toxin in the blood that could get out and leak out and kill this neuron. Well, it will get blocked by the astrocytes before it gets to the neuron. So interestingly, there are a lot of drugs that help target certain neurodegenerative disorders like Parkinson's, Alzheimer's. There are certain drugs that have been proven to do good work for those diseases, but the problem is figuring out how to transport them past that blood-brain barrier is proving to be pretty difficult because they are very selective. Now, throughout the brain and the spinal cord, you'll also have basically this fluid that's bathing these neurons, and this is called cerebrospinal fluid, and it is going to be secreted by another glial cell that'll be lining the ventricles of the brain and this or the brain primarily. And these are called ependymal cells. Ependymal cells. And these guys, again, they look kind of cuboidal. They will secrete that CSF. So those are all the glial cells of the nervous tissue in the central nervous system. Now, the peripheral nervous system only has two. They've got satellite cells. Okay, satellite just translates to they're kind of like around the rest of the neuron. And I just think of these as the do-it-all of the peripheral nervous system. They're going to help protect it. They're going to kind of cover the axon. No, not the axons. They're going to basically nourish it, respond to inflammation, all that. So it's a grouping of several different functions. And then we will also have our Schwann cells. And the Schwann cells are going to form, again, that myelin sheath. So say we've got our neuron here. The Schwann cells of the peripheral nervous system, again in your nerves, are going to form the myelin sheath. And it actually looks a little different. They kind of look like this and then they wrap it around it. Okay, so I'll just draw it like that. Okay, so they'll form that myelin sheath, the Schwann cells will, on the peripheral nervous system axons. And again, these are insulated myelin. They are only in the white matter. So if you learn about gray matter, white matter is essentially all the myelinated axons traveling up and down and out of your nerves, as well as up and down your brain and spinal cord. Whereas if you don't have those, if you don't have the myelin, it's actually gray matter. This is where a bunch of neuron somas, cell bodies, are really close together, just kind of interacting in a very small space. It's kind of like a huddle in a football game. Okay. All right, y'all. If there's nothing else, I think that's it. So, hey, again, thanks for joining me on this live stream. If it's been helpful, please check out the other videos. We got a lot of them about these topics that I've already taught. So please let me know if you have any other questions, and I'll let you know as soon as we do another session of this. In the meantime, sign up for some tutoring with my Calendly invite in the description. And again, thanks for watching, guys. It's been a blast hanging out with you for this hour. Happy Sunday, and best of luck on all your tests, all your healthcare. And can I just pray for you real quick? Lord Jesus, I pray whoever is watching this right now would just come to know your grace, your love, your truth. And Lord, it is such a privilege to be even able to study the human body and all of its amazing, amazing complexities that you have made as you've written in Psalm 139, verse 13 through 16. Lord, we are fearfully and wonderfully made. I pray that every person who watches this and hears this knows that you love them so dearly, so much that you gave your only son to die on a cross for their sins so that they can be in right standing with you, be raised up in this life as well as in the next in eternity. So Lord, we thank you. We pray that this would bless them. Whatever I've taught, Lord, I pray that it can stick, that they can study well, work hard, and be successful healthcare practitioners, and bless this broken world for your kingdom, your glory. We pray these things in Jesus' name. Amen. Hey, thanks for watching. Take care. If I can turn this off properly, we'll find out.